I'm Umar Ali. In this video I will talk about plant tissue culture techniques. So guys, if you haven't subscribed to my channel, please subscribe and click bell so you will get notification when I upload a new video. So starting the video. The plant tissue culture, it is a method in which we can grow a whole plant in laboratory or in vitro by using single tissue. It is in vitro propagation. We can grow the whole organism or plant in the laboratory. It is rapid as compared to the outdoor, and it is asexual. The rapid, if we want to grow a plant with the foreign gene, the growth will with ordinary method will be slower because it has to take a lot of time, 2 to 6 months, but this is a rapid process. It can take 4 to 6 weeks, and the new plantlet can be easily grown. The genetic material of the parent plant will be retained. It will build the exact replica copy of the parent plant. The plant tissue culture can be used for the micropropagation as well as regeneration. So discuss it led by one. The vegetative propagation occurs in nature. The layering or dropping lower branch contact the soil. It is pressed down by snow or by vegetation. The roots form at a point of soil contact, forming a new genetically identical tree. When trees of some species are cut down, the shoot emerge from the stump. Strawberries spread through, sending out above ground horizontal shoot. Cardinals, also called stolons. There are other examples you can learn from internet. The micropropagation. Many genetically identical plants are produced from one plant. It is in vitro propagation in tubes or petri plates. It is rapid and asexual as well. A predator plant is superior. It has higher, a good moisture and starch content, and good for French fries. Isn't it? Micropropagation. A single node will produce a shoot from 4 to 6 weeks that has 4 to 6 nodes. The next these 4 to 6 nodes can also be used to propagate other plantlets. So each plantlet can be subcultured to produce another 4 to 6 plants. Hundreds and thousands of plants could be developed from one node. Since these are produced from the auxiliary bud, plantlet will be the clone of the mother plant. The genetic material will be the exact replica of the parent plant. Micropropagation of almost all the fruit crops and vegetables is possible. For example, dwarfing sweet cherry, shade tree, ornamental shrubs, roses, calamitous, lilacs, etc. Rice, cotton, tomato, potato, citrus, turf, legumes, etc. Advantages of micropropagation. It is economical in time and space. The greater output can produce millions of the uniformly flowering and yielding plant. African biotechnologies, fruit crop, banana, and indoor pot flower 6 million pieces per year. Disease-free elite plant with exceptional characteristics. Advantages: continued facilities. Facilitates safer movement of germplasm across nation. In vitro germplasm assures the exchange of pest and disease-free material. Great for vegetatively reproduced crops, crops which produce few seeds or high heterozygous seeds. Regeneration: the process whereby a part of plant can be turned into a whole new plant. African violet leaf sprouting new shoots by the regeneration process. Regeneration is possible because plant cells can be made totipotent using hormones. Differentiated tissues like stems, leaves, roots, etc. Undifferentiated tissues, embryonic cells, are totipotent, can become a whole new plant by differentiating into a whole new plant. The plant hormones are naturally occurring chemicals that influence plant growth. Growth regulators are synthetic versions of hormones. Cytokinins induce the production of shoots. Auxin induce the production of roots. The action of different hormone regulators is not consistent, and different plant will respond to the same chemical differently. Different plant parts from the same plant can respond differently, but we can use these hormones for the growth or regeneration process. Steps involved in tissue regeneration. One of the important aspects: tissue must be sterile, completely free of any microorganism, done using a aseptic technique. Number one, starting tissue is called an explant. Differentiated cells. These cells have developed to be part of specialized tissue, root, leaf, stem, ovary, cartilage, etc. Explant of the plants are on a sterile petri dish containing hormones and nutrients that promote the explant cell to develop into a mass of undifferentiated cells. Callous cells are totipotent. This is the tobacco regeneration in vitro. The third step is the individual cell or clump of cells of the callus are transferred aseptically to different petri dishes containing sterile medium that encourages the undifferentiated callus cell to become shoot and roots. So one mass of callus cell can be divided and transferred to the many plates for development into shoots and roots. You can see that in the diagram. Once shoot and roots have developed, they are transferred to soil and grown to maturity. This is the diagrammatic explanation of regeneration by the tissue culture. So I will explain it to you. This is our parent plant. We can wash carrot, tap root and make it aseptically free of any microorganism. Break into 3 or 4 pieces. After that, insert a cork board. The region where cores are be taken. After that, cut transversely into sterile petri dishes. Then place dishes on the callus initiation medium. These media can help the growth of callus. Callus formation is occurring in 4 to 6 weeks. This is the callus formation. After that, it is transferred to root or shoot development medium. The root and shoot development medium, root and shoot development to 4 to 8 weeks. Then it is transferred to the soil. This is optional. Then it leads to the growth of a mature plant. This mature plant can be used to for the production of other 5 to 10 plants. So it is a very easy technique. It can be done by a little expertise, and it is efficient, fast method. Micropropagation versus regeneration. Plants that develop from existing terminal or axillary bud are true clone of the mother plant. The mesmetic bud tend to be protected from the variation. Sorry for the inconvenience. Mesmetic bud tend to be protected from variation caused by environmental factors. Little or no variation usually occur when this method of tissue culture is used. Okay. The regeneration using callus can produce a variant of the original plant, but non-mesmetic cells are not protected from the variation caused by the environment. When these cells experience a mutation, the plants that are produced from them will also show a difference from the mother plant. This is one of the major difference. Tissue culture also used when plants are genetically engineered. Plants that receive a new gene are called transgenic plant or GMOs. A genetically engineered plant has a new gene squash gets caught protein from the cucumber or zucchini virus to prevent viral disease. To vaccinate the squash, corns get a gene from the bacteria that allow it to survive in the presence of weed killer, one that is less harmful to the environment that other herbicide. Corn gets a gene from the different bacteria that allow it to produce its own pesticide product against insects. After a new gene is given to the plant, scientists must figure out how to regenerate the transformed plant cell. So that's all from today's lecture. So guys, if you have learned, please like my video, share it to your friends, and subscribe and click the bell. Thanks.